I want to welcome all participants on behalf of the ESDR. Many of you will know of the kitchen webinars. They come in 4 themes or flavors, and this is the sweet and sour theme, in which a particular topic will be discussed and sometimes even debated. We have 2 excellent speakers who will exchange ideas about the function of the skin microbiome and how to investigate the skin microbiome. This particular webinar is sponsored by BMS. Before my co-chair, Rajan, will introduce the first speaker, I would like everyone to invite placing their questions in the chat functions of Zoom, so we can incorporate your questions in the discussion that will be held after the 2 10-minute presentations.
Thank you, Remco, and it's a great privilege to have our sweet and sour kitchen today with the topic of skin microbiome from association to function. As you know, we have 2 world leaders who will then debate this in 2 10-minute blocks. And it gives me great pleasure to introduce Professor John Comyn, who's Professor of Cutaneous Inflammation and Microbiome at Newcastle University, as well as Deputy Executive Director at A-Star Skin Research Labs. And John started his research career following a postdoc at Queen Mary University, London, and then he's been in A-Star and has been rising there and was promoted to PI in 2013 in the lead up to now being Deputy Executive Director. He works in the area of skin barrier and immunopathogenesis and the microbiome, and he established the Singapore National Atopic Dermatitis Program and the Asian Skin Microbiome Program, and is keen to catalyze and promote research in atopic dermatitis and the skin microbiome. So John, without further ado, please, the floor is yours. We invite you to share your slides and open the debate on skin microbiome from associations to functions.
Oh, great. Thank you, Neil. Thanks for the invite, and happy to do this today. Hopefully everyone can see my slides. So indeed, we'll talk about the skin microbiome today, and from associations to functions. So just as a way of introduction, we've been studying the skin microbiome for quite a while now, and it really started to take off with an explosion of microbiome research in 2008 on the back of the microbiome program in the US. And it's continued, and the publications have grown over the years at quite an extraordinary rate. And what we can see is that although the microbiome is well studied, the skin microbiome is still quite a small niche of research. When you do a PubMed search for microbiome plus skin, there's still the numbers really do dwarf the total number of microbiome studies. And if you include something like fungi into the search as well, and the numbers drop even further. However, this shouldn't be the case because, of course, skin is a major epithelial surface. And if we look at it as a flat surface, it's about 2 meters squared. But actually, with all of the various
I think John's connection has frozen briefly, but I'm hoping he will rejoin us in a moment.
Bodies changes in. Sorry, can you hear me, Neil?
Yes, we're good. You froze for about 15 seconds.
Okay. I was just saying that the skin has a unique and variable ecosystem. The humidity, the temperature, the pH, the composition of AMPs and lipids across the body surface can change, and this can has a profound impact on the skin microbiome across the body. And when we think about culturing microbes off the skin, this really only gives us a partial view of what's present. And so many studies have now moved towards a next generation way of analyzing what's present on the skin, and move towards genomic approaches that reveals much greater diversity of the microbes than what we were able to see with culturing. And there's been a number of different approaches to do this. And one that we've employed quite a lot is shotgun metagenomics, to study the whole microbiome, all of the genes, all of the genomes, rather than using marker genes such as 16S. And I think if you think about this approach, it allows you to do not just the taxonomic profiling from the reads, but also allow you to look deeper into the genomes for the virulence factors, as well as trying to drill down into more functional profiling. However, there's not really that many publications on skin shotgun metagenomics, and so there's still a lot of potential for growth in this area. And I think there'll be many more studies to come as the cost of sequencing goes down in the future. What is known is that across the body, as mentioned, these different body sites, which are shaped by our physiological characteristics, give rise to different compositions of the microbiome. And these are generally classified into the sebaceous sites, the moist sites, and the dry sites. And you can see that the proportions of the kingdoms and the fungi and the bacteria are altered across these different body sites. And if we look within these particular body sites, what we can see, say if we look at the antecubital fossa, is that the intrasite variability is greater than the inter-individual variability. So in this study here, we can see 4 individuals have rather similar profiles when we look at the bacteria at the antecubital fossa, as comparing to the back, the nair, or the plantar heel, which have very divergent microbiomes compared with the other body sites. So thinking about different body sites is very important. In our lab, we've been studying atopic dermatitis, and this is a chronic inflammatory skin disease characterized by dry, itchy skin with recurrent lesions. And the pathogenesis of AD is quite complicated, with a component of skin barrier dysfunction, a strong impact from the immune system, as well as the contribution from the environment and the microbes. And this gives to a very heterogeneous disease with many different endotypes. And many people are interested in what's the role of the environment and the microbes in this situation. And so the microbes do play a major role. We can see that about 3% are predisposed to herpes simplex infections, and around 90% are colonized by Staph aureus. But why this susceptibility is occurring, it's unclear, and why the different microbes change over time is something that's been heavily studied over the years. And so Heidi Kong, with a seminal paper in 2012, showed that across the atopic dermatitis flare cycle, you can see significant changes in microbial diversity from the blue line, and changes in the Staphylococcal aureus, as well as changes in the disease severity as the microbiomes tracked over this time, from the baseline across the flare and into the resolving flare and the post-flare times. And this shows that there's really a dynamic microbiome alterations across this flare cycle, which is very interesting to study. And so for us, we decided to look in the non-flare situation, and what we could see is by looking at various different people, we're able to see different clusters of patients that dropped out. So we called this red cluster that you can see here dermatype B, and then this blue cluster that had a mixture of atopic dermatitis and healthy controls, dermatype A. And interestingly, this dermatype B, as we called it, had a higher presence of Staph aureus virulence genes when we looked in the microbiome, because we were doing the shotgun metagenomics, we were able to see this. Other aspects of this kind of study that you can do, you can drill down into the functional profiles. And when we looked again into the genomes of the microbes that were higher in the AD cases, we could see increased metabolism of arginine, nitrogen, and proline, which could be associated with ammonia production and elevated pH. And on the other flip side of things, we could see that there was lower pathways related to tryptophan metabolism, which predicts that these microbiomes would have less tolerance, immune tolerance, alterations in the barrier function that could be related to AHR signaling. So by looking at the microbiome, we can actually find out a lot about the skin and about the host microbe interactions. And in this study that I told you about, flare susceptibility related to microbiome signatures, we can see that there are distinct changes in the non-flare skin related to the taxonomy, an increase in Staphylococcus aureus virulence factors. We can also see that the skin microbiome signatures can help to stratify patients. We see increased severity in some of these endotypes. We see increased flare episodes. And we can also see that these microbiome signatures could help distinguish AD endotypes with higher IgE, increased AMPs, and a change in the Th2 immunity, which I didn't show today, but we're all able to see in these patients. And so I think looking at the genomes, looking at the genetics of the microbiome is very interesting. It allows us to think about therapeutic interventions that can be built around the microbiome. Can we target the immune axis and alter the microbiome, which I think people have now done by treating with things like dupilumab and atopic dermatitis. We can see changes in the microbiome. There's also people that are thinking about, can we now use this information to transplant microbes and microbiomes that are missing from our skin microbiome to change the inflammatory situation, and also use microbiome to induce epithelial repair. So I think looking at the genomes, thinking about what's on humans has a lot of potential for our understanding about our host microbe interactions. And with that, I will leave it there and hand it on to the next speaker.
Wonderful, John. Thank you. That's been fantastic. And Ellen, you're about to start, and Remco's just joined us, and I'll allow him to introduce you.
Right. So thank you, John, for this clear presentation. You focused more on the omics side, and Ellen is a bit more for the functional side of the microbiome research. So Ellen van den Bogaard is professor in experimental and translational dermatology, and also head of the experimental dermatology lab at Radboud University in Nijmegen, in the Netherlands, where she also studied biomedical sciences. And after her PhD studies there, she did a research fellowship at Pennsylvania State University, working on the target for coal tar therapy in inflammatory skin disease and the aryl hydrocarbon receptor signaling. Well, in her lab, she designs or engineers 3D organotypic skin disease models, and also studies the interaction between the constituent skin cells and the skin microbiome.
What I would like to make a strong case for is that next to studies into the genomics of the microbiome and looking into patients or healthy volunteers and looking at the associations of these microbial compositions to either health or disease, that we should couple these to actual functional studies to investigate what the buggers that live on our skin actually do to our skin and how can we intervene. So what we always propose is that there should be this research cycle that goes from observations in patients or in healthy subjects, like the studies that John already mentioned, sample this microbiota and identify these microbial suspects that are either related to health or disease or to severity, for example. However, we try to take this information into our lab and to pinpoint what these changes in the microbial composition actually matter, and also whether or not this information could help us in devising intervention modes for patients. And I think both John and I, the studies that we do, we always have a focus on generating impact for healthcare and either trying to understand disease better, trying to find biomarkers that could stratify patients, for example, but also to see whether this information could lead to better healthcare. So what I would want to focus on today is in this cycle from association to causality and its actual application of the data, that we develop models, select specific microbiota that are either overrepresented or underrepresented on skin, and actually functionally analyze these candidate microbes by using these skin models that we have in the lab. And I want to show you some examples in our lab, but also in that of others. So I think typically, traditionally, a lot of studies, if you look at the microbiome, they're still focusing on animal studies, which I think have told us a great deal, but can also not only be the only model to study the skin microbiome, and especially if we want to look at the human microbiome. So typically, mice, either pathogen-free, specific pathogen-free, or germ-free mice are used in these studies, either are not complemented with antibiotics treatment to then see what specific application of commensals or pathogens on the skin of these mice have an effect. So either microbial transplantation or looking at either infection or colonization of the microbes on these mouse models. I think one nice paper that I would like to highlight here is the paper by Uberoy from the group from Elizabeth Grice, published in Cell Host Microbe, where they use these different mouse models to look at what commensal microbes from the human skin microbiome have for an effect on the skin barrier. actually identifying the aryl hydrocarbon receptor as one of the keratinocytes receptors that mediates this host microbe crosstalk and that actually regulates skin barrier function in these mice. And I think we all know the work by Richard Gallo's lab, also looking at microbe-microbe interactions, so between aureus and epidermidis, for example, or hominis, and also looking there for potential probiotics that could help to the skin health. But I think we all recognize that there's a growing need and growing demand for using human models in skin microbiome research. To either move away from animal studies or to complement the animal studies that are already currently out there. And I think there's different flavors that can that we can utilize. Obviously, using skin from healthy volunteers, using ex vivo skin cell cultures, but also the organotypic skin models that I will come back to in the next slides have been greatly instrumental. And there's different ways into studying the host microbe interactions, either by selecting specific pathogen-associated molecular patterns, the PAMPs, using heat-killed lysates, live microbes, and also here there's always the delicate balance between looking at infection versus the true colonization of these models by the microbes. And often this colonization will be followed up by infection. So also here looking at time frames and whether this is truly a host microbe interaction resulting from colonization or whether you're already looking at an epidermal infection is quite key. So there's different ways to do this, either with the organotypics, as I mentioned, using full thickness skin equivalents or epidermal equivalents. And I think recently the models from the induced pluripotent stem cells are actually hair-bearing and having the appendages could be instrumental for these kind of studies. So some things to take into consideration when you want to use these organotypics. This is a study by Wu et al. Looking at MRSA and Pseudomonas aeruginosa, basically inoculating skin equivalents, in this case derived from entered keratinocytes, by the microbes, and then also looking at the formation of biofilm formation and some antimicrobial peptides, this DGK that you see here, on the formation of the biofilm. And here, both the effect on the biofilm, but also the effect on the epidermis can be studied in this model where you have viable microbes, viable keratinocytes, and where you can neatly study this. So these host microbe interactions can be neatly studied. However, we then depend on having culturable microbes. As John already mentioned, our skin is full with microbes that may not be culturable in the lab. These skin equivalents cell cultures often are in aerobic culture conditions and in humid culture conditions. These typically only model the interfollicular epidermis, and there's quite a high risk of contaminations when using these cell culture-based method. And there's a nice letter in the Journal of Investigative Dermatology by the group from Julia O, also looking at how different models, for example, epiderm, lab skin, or native skin that you see on the lower right, actually have a different effect on the microbial composition when cultured with these different models. So also here, the type of model that you use greatly influences the outcome. This is something to bear in mind when you want to do these kind of studies. So what we've tried to do is to develop better systems to apply these bacteria, making sure that we lower the risk of contamination because of the edges of these organotypics are always less well formed. And you often have contamination through the edges of these organotypics. So we use the glass cloning cylinder for a large application area of the bacteria, meaning that you can apply a large volume of bacteria and have a large surface area covered by the bacteria. This paper published in Microbiome last year by Gijs Rick, a PhD student in our lab, where we also looked at dry versus humid culture environments, looking at 37 versus 32 degrees, which is the latter is obviously the temperature of our skin. We could demonstrate that we could co-culture these bacteria up to 2 weeks, but there's great dependencies on the strain that you use. So different Staphylococcus aureus strains result in different effect sizes. So there's a great care to take into consideration when you want to study this in the lab, that you would always have to take multiple strains into consideration. And there's multiple readout parameters to really study this functional effects of having a microbe introduced into your cell culture. Obviously, these are all single microbial cultures, and the real deal should be that we apply full microbiomes onto these models to study the composition. And this is what we've done with these organotypics, and sampling them over 7 days of co-culture, and then looking at the diversity, which definitely changes, but also still greatly represents the diversity that we see on the lower back of the healthy volunteers. And I just want to make one case of how these models can be used to study the functional consequences, also relating to the work that John already presented on atopic dermatitis. And we had the question how loss of filaggrin would impact the skin microbiome. So a lot of focus is obviously on Staphylococcus aureus, but we wanted to know more about specific commensals that may be lost when you have a lack of filaggrin in your skin. So we did a microbiome study. This was one 16S. And what we found was that there was a specific commensal group of bacteria, gram-positive anaerobic cocci, that were less presented on the patient's skin, in this case, Ichthyosis vulgaris. We wanted to know whether this was a bystander effect or really an effector that could drive the disease. And the paper by Van der Krieke et al. In iScience nicely shows the myriad of experiments that you could utilize to go from this association to function. This is just a panel to represent what, in this case, Danique did, all of the different cell cultures, looking at AHR signaling, for example, looking at antimicrobial proteins and that barrier function when skin is exposed to these gram-positive anaerobic cocci. And I think really the complementary experimental setup should always be taken into account, because not one model can definitely represent the full skin and its interactions. And what Danique was able to find was that these gram-positive anaerobic cocci that we miss on the skin of Ichthyosis vulgaris and atopic dermatitis patients are actually quite key in induction of antimicrobial protein expression. They induce a host defense quite quickly and also induce epidermal differentiation. And we took it actually one step further, is that we fractionated these bacteria to find the active. So what are the active metabolites in these that are produced by these bacteria? We found them to be indoles and to be activating the allyl hydrocarbon receptor, like coal tar is doing and like tapinarof is doing, and also what has been demonstrated by the paper from Elizabeth Grice's cell host microbe. So therefore, we could suggest from these data is that this could open a door for microbiome-directed therapeutics. So if you want to activate the receptor, instead of using coal tar or tapinarof, maybe we want to change the microbiome into having commensals there that could activate this receptor and fostering skin health. And with the last slide I would want to end is that the models that we are currently using, these organotypics of the interfollicular epidermis, are just one model to do it. And I think the future is quite bright for these human model system, as we now have these appendage-rich organoids from the Carl Köhler lab. And recently also in iScience, Jung et al. Showed that these could be used for bacterial colonization. And this could all be nicely complemented by in silico approaches, like from the group from Reiko Tanaka, demonstrated that these experimental data helped to feed these in silico models, where we could predict how differences in the microbiome could actually change the status of the skin. So with that, I would like to add in saying that next to these genomic approaches and looking at patient skin and the patient microbiome, that these should be complemented by functional studies if we really want to move from association to its actual application and intervention. Thank you.
Thank you very much, Professor van den Bogaard. I would like to again invite all the participants to post their questions in the chat function. This is your opportunity to ask the leaders in this field of research. And maybe to start the discussion, it appeared to me that your approaches to studying the skin microbiome is in a way complementary. It's not that there's a real contrast in the way that you would view how this type of research should be performed. More for John, I was thinking with regard to your atopic dermatitis flares. Have you also performed longitudinal studies in which you have multiple time points, and could you perhaps see if the flare was preceded by a certain shift in the microbiome? This would allow you perhaps to disentangle this cause or consequence a bit more.
Yeah, so we didn't in the studies that we published, but we currently have some ongoing studies to try and track this. That's, I think, a very relevant question. It's definitely what needs to be done, more temporal studies to understand what happens during the life course of a disease.
Thanks, John. Remco, if I can also then follow on with that with a brief mention of Ellen, those models that you showed were fantastic, and I was curious if people had started to compare, what microbes are sustainable in 2D cultures and conventional 3D skin equivalent cultures compared to organoids, which have hair follicles and have got sebocytes, producing all the goodies that microbes like on human skin. And have people started to compare, the taxonomies across models to see which might be closest to human skin?
Fantastic question. I think the one study that have tried to do this, but only for the interfollicular organotypics, was a study from Julia O published in the JID, actually showing already within the interfollicular epidermal models that there's differences. But I can only imagine that with the organoids, it will greatly start to differentiate even more. We haven't done such studies also because specific models are used in specific labs. So that also requires collaboration across the different labs, which I think should be done. And also, I think currently we're having a cost action, NetSkin models, that also fosters such cross collaboration. But the only thing that I would also want to mention is that the microbes that we add onto these organotypics, they feed from the stratum corneum, right, and the proteins that are there. So also it greatly differs in the way that this stratum corneum is built up. And for example, the hair-bearing organoids from Carl Curtis' lab, if you have a keen eye for the interfollicular epidermis, that epidermis is actually not so nicely formed. So the appendages are super cool, but the interfollicular epidermis is not so nicely formed as that we typically have in the other models. So there I could also envision that you have differences. So, like I said, models should be complementary to each other and should be validated, either with the human data from John or with whatever other models that are out there, because only one model should not tell the whole story.
Maybe a question.
Just an answering to the question in the chat, we don't have the immune cells yet in the models, but there's models that do have these, either the Langerhans cells or the dendritic cells, and currently working on making these immunocompetent. But this is definitely something that should be taken into consideration. Now we consider the keratinocytes being our innate immune cells, which is obviously a bit of a simplistic way of thinking. And even the fibroblasts could have like an additional cellular interaction there. So there's, let's say, the future is bright and much more to study.
A question that I would like to read from the chat by Dr. Patra. For John, do you have any comment on fungal communities and their role in atopic dermatitis?
Yeah, well, we do see the fungal and we profile it during the metagenomics, and we do see shifts in the patterns. In terms of what I like to comment, I mean, they're very important. The fungal communities are very active. There are ongoing in vitro studies that I think both Ellen and I are probably aware of that are looking using models.
You freeze.
It's unfair in a debate when the technology counts against you.
I guess what John was referring to were the Malassezia studies, where different strains of Malassezia also induce differences in the organotypic models, but the Malassezia are quite difficult to culture.
So John, you're getting a collaborative support from your debate partner here, which is fantastic and very collegiate. So thank you both. There are many fantastic questions in the chat. If I may also read one, and it's from Michelle Simon, and it is: what is the relationship between skin and gut microbiome, and could we modify reverse altered skin microbiome acting on the gut microbiome?
Yeah, I think there's a lot more work to be done here. There's just in short, just to keep it short, there's emergent studies that shown very strong connections between the two, and I think there's going to be a lot more of research in this area.
And maybe one to add to this, a question by Sarah Brown that I think is very relevant. If you both could comment on the feasibility of the changing of an individual skin microbiome, because it may have the tendency to revert to the previous state.
Yeah, I agree. I think that's coming out from some of the.
Ellen, maybe you need to take over as he is frozen.
No,
so
John will come back. John will come back. I think I don't want to.
It's a 10-second thing, I think. I know John is not at Newcastle at the moment. He's on tour.
Yeah.
Sorry.
John is back.
Sorry, John. We missed the answer. You only just started. Would you care to repeat?
Yeah, I was saying that I think immunotherapies are able to change it, but that seem to be it seems to revert back to normal, right? So I think people have a baseline microbiome that seems to stick with them. So it's difficult, I guess, to modify forever, but I guess that does provide many opportunities as well.
Perfect. Thanks, John. So one question for Ellen from Franz Hilke. Hi, Ellen. In your presentation, it looked like a large proportion of the microbes are lost during transplantation of the microbiome. Can you tell me more about which microbes don't survive the culture and why? And then maybe there's a related comment from Yves Poumet about would be nice to culture Malassezia with cephalocytes containing model indeed. So Ellen, if you wouldn't mind touching on those.
Yeah, so I think Frans is referring to the slide where we had the microbiome, full microbiome swap on the 3D culture, and typically what we see is that the aerobe bacteria they like to take over. These are staphylococci, obviously, and they sort of occupy the niche and occupy the space on the stratum corneum. And that bacteria like Cutibacterium acnes, for example, that typically reside more in the follicles, that they can survive, but they will not grow. So I think what you're looking at, and that's what the thing is with these analyses, is that it's relative, right? So this was 16S, so it doesn't tell you also about viability, right? It just tells you about relative composition. So this is also something to bear in mind in doing such analyses, is that we would also be quite keen into combining either culture with sequencing, because the bacteria also might just be dead, but you'll still pick the DNA up, and hence you're then seeing it back into your results. But definitely the staphylococci, when we do the culturing, the staphylococci seem to prevail. And also that is why we started to change cell culture conditions regarding temperature and humidity, because with lowering humidity and lowering temperature, you can control the system better, because it favors the staphylococci a bit less. So you're leaving more space for the rest.
Perfect. Thank you. Remco.
Yeah, maybe a more clinical question. So for atopic dermatitis, the treatment with antibiotics is generally not very effective. Is this related to the fact that the bacteria that are causally involved in the disease process are not eliminated by the antibiotics that are normally used, like penicillins? Or is there another reason why they're not so effective? Because this gets at the cause or consequence question, of course.
Yeah, I guess if I would answer this from our view, that what we've learned about the commensals, and specifically, for example, the gram-positive anaerobic cocci, is that if you would eradicate those, that would be sort of detrimental for sort of your antimicrobial shield, and also potentially for barrier function, since we know that these microbes are also instructing the skin into either epidermal differentiation, lipid processing, etc. So if you sort of kill them all, that would be on the short term clearing infection, but on the long term you would miss sort of the microbial cues that could help the skin in maintaining a better barrier or maintaining a better antimicrobial profile. So,
John, would you like to add to that?
Yeah, I think I agree, right? It's sledgehammers are going to take out a lot of the microbiome, so we need more precision with the tools that we're going to use for teasing apart the microbiome.
Neil, I see that we've exceeded our allotted time already by a few minutes. I'm not sure if there's room for more questions, or if we should close the session.
I'm afraid we might need to close, actually. I mean, we've had a fantastic turnout.
Yeah.
I think we have more than 80 people on the call, but some people are already starting to have to go to their next meetings. So Leah is going to share some slides, and Remco, I'll let you promote our next upcoming opportunity.
Right. Well, before doing so, I would again like to sincerely thank our esteemed speakers.
Yeah.
It's been great. So Ellen van der Bogaard and John Commons, thank you very much on behalf of the ESDR and all the participants. We've learned a lot about the microbiome, and it was nice to see also that your views are complementary. So it was not a real debate, but you seem to agree on this. Well, I would like to draw your attention to the next kitchen webinar session, which will be on micro needle systems for the enhanced management of skin conditions, as you can view on the slide. It will be given by Ryan Donnelly on November 6th. I would like to thank the sponsors again, and also all participants in this kitchen webinar. I hope you've enjoyed it all, and it can also be viewed on the ESDR website, so you can view it again if you like, or share it with your colleagues. Thank you so much, and also thank you, Neil, for co-chairing. I'm not sure if you would like to give another
Thank you for a great dialogue and discussion, and as I say, fantastic feedback in the Q&A where people are both complimenting both of the speakers. So thank you so much, and yes, bye everyone, and take care.